So this section in the chapter on photosynthesis focuses on pigments. And so our learning outcomes for this one include discussing how pigments are important in biological systems, and number 2, relate the absorption spectrum, which is something we dealt with in the spectrophotometry lab. So you're supposed to relate the absorption spectrum of a pigment to the color that the pigment produces. So what are pigments as a vocabulary word? These are going to be molecules that absorb light within the visible range, within the range of electromagnetic radiation that we can see. And light, generally light is a form of energy. It's a form of radiant energy. It can be called radiant energy or EM for electromagnetic radiation. Physicists have described light as behaving as both wave and particle. And the particle behavior of light is called photons. So the photons are the particles of light, and they're going to act as discrete individual bundles of energy. And the energy content of a photon, as we're going to see in the diagram here in a bit, is inversely proportional to wavelength. So one of the things that gives us different colors is by changing the wavelength. Longer wavelengths, and then you get shorter wavelengths. So you can start off on the order of 100 meters from here to here, and then end up in the nanometers, which is really tiny. So what this is saying here is the inversely relationship says that as the wavelength gets smaller, then the energy of the photon gets bigger. So the smaller the wavelength over here, greater the energy of those photons. The longer the wavelength, the lower the energy. So over here you would have things like X-rays and gamma rays, and over here you would have like radio waves. We'll see this in the diagram here in a bit. And then somewhere there in the middle is where you're going to have visible light, very small portion of it. So anyhow, these photons or these packets of energy can actually produce what's called the photoelectric effect, which is when a photon strikes an atom or a molecule at a very certain specific wavelength or a very specific amount of energy, can actually remove the electron from the atom or molecule. And then that moving charged particle, that electron, can be used to do work. In a calculator, those electrons, using a solar cell, a calculator, like this calculator I have right here, there's a solar cell there right at the top. The photoelectric effect would remove electrons from the surface of that material and then get them to move through a circuit and can do the electrical work to drive the calculator. In the case of plants, the electron can be used to do the work of producing ATP and NADPH and ultimately producing organic molecules from that chemical energy. So the photoelectric effect takes our radiant energy and allows us to convert it to chemical energy. So here's your electromagnetic spectrum, and you can see it has the entire range from left to right here. And you can see right there towards the middle, you can see the rainbow is a very small portion of the entire electromagnetic spectrum. The part that we can see, or that we have the tools to detect within our eyes, is called the visible spectrum. And a good way to remember it is to say, and we're going to have to go backwards here because of the way the spectrum is represented here, is red or orange, yellow, green, blue, indigo, and violet. Or in other words, if going backwards, you're saying Roy, G, Biv. And the reds are going to have the longer wavelengths. You can see here 740 nanometers in size to 400, which is actually smaller nanometers in size. And you look up here, you can see that one of the scales has the wavelength and the other one has energy. And you can see they go in opposite directions, increasing wavelength moving to the right, but increasing energy moving to the left. So that shows your inverse proportional relationship between the two. You can see your longest waves out here. We can't see them. Those are your radio waves. These include microwaves, which power your cell phones. And then we have infrared. Infrareds are just below the red range, and some of the infrareds you might be familiar with keeping your french fries warm at the fast food restaurant. Ultraviolet light we cannot see. It's very high energy, good enough to give you sunburns, damage molecules. X-rays even more so. which we can also use to see bones within the body, and then gamma rays, and then you can go on to cosmic rays, which all are more and more higher energy way out there. So what is an absorption spectrum? This is another vocabulary word. This is going to be looking at the range. So looking at the definition down here for absorption spectrum, this is going to be the range at which the photons striking a molecule are capable of being absorbed. So we're going to look at the entire range for a substance or a molecule, and we want to study what types of wavelengths are these molecules or substances most capable of absorbing. In other words, what are the right energies? What are the right wavelengths that these specific molecules interact? And if we see that different substances are going to interact differently with electromagnetic radiation, so they would produce different absorption spectrums, almost like a fingerprint. And so we'll see some absorption spectrums for some different pigments found in plants and see how they look different. They're like fingerprints for them. And here's what happens when photons strike a molecule. The energy can either be lost as heat, or it can be absorbed by those electrons in the molecule. And when it gets absorbed, it boosts those electrons into higher energy levels. If nothing happens to those electrons, then they come back down to a more stable state, and then they re-radiate this energy either in the same or different forms. So it may come out back as the same color, or it may come out as some different color, or even outside the range of what we can see. Maybe given off as just infrared heat. It just depends on the molecule and the photons that are striking those molecules. So here we see an absorption spectrum. If you haven't already had a lab, you'll have a lab where we use a spectrophotometer to measure absorption spectrums of different substances. And here you can see there's different substances on here. On the bottom axis we have the wavelength that we're measuring of the substance. So usually these pigments will be in solution, and then we'll put light going through the solution and then measure how much of that light is absorbed under different wavelengths. So we might go at 400 and then 425 and different wavelengths, and then make our measurements. How much of that light is being absorbed? There's a machine that can do that called a spectrophotometer. And you can see that the solid line represents chlorophyll A, according to our key here. The dotted line right here represents chlorophyll B, and then the gray line represents the carotenoids. So you can see each one of them has a different absorption spectrum. So if I were to ask you at what wavelengths does chlorophyll A absorb best, I would look right here at this peak where we have the highest absorption for it, and then I can come down here on the scale down here and estimate from the graph, maybe around in the 420s, 430s that it does so. Notice how chlorophyll B has a different optimum absorbing wavelength of light, and you can see that for chlorophyll anyway, it has two major peaks. The other one is over here out at somewhere between 650 and 700, maybe about 670 or so. And so right now we get a sense of why maybe plants may look green to us. If you shine white light at plants and they're absorbing lots of the blues and the violets over here and lots of the reds, well, what does that leave? That's going to leave only green light to bounce off the plant or pass through our solution of chlorophyll, because the other wavelengths are being absorbed. So white light has them all the colors of the rainbow. So when you shine white light at a plant, it only gives off, as you can see right here, what you end up seeing is green, because that's what's not being absorbed. So pigments in photosynthesis, which we saw a graph earlier, some of the major ones. Some of the pigments, of course, they've evolved over time. Even we have pigments, they're not used for photosynthesis. We have pigments like melanin, which help protect us from ultraviolet light, gives our skin some color. But these pigments have evolved over time. As far as plants go, the pigments for photosynthesis, specifically right here, are two general types, the chlorophylls and the carotenoids, and we just saw the absorption spectrum for some of those pigments. Some other photosynthetic organisms are going to have other molecules to help absorb light. We're going to see a couple of examples of those right now. So looking at two major types of chlorophyll, we have chlorophyll A and then chlorophyll B. Chlorophyll A is going to be the main pigment in plants and in cyanobacteria for driving photosynthesis. And the reason for that is that this is the only pigment that can be used directly to convert light energy to chemical energy. That is, it's directly involved in chlorophyll A in making both the NADPH and the ATP, which ultimately help make our organic molecules, which is where we're going to store the chemical energy. And as we saw in the prior slide, the chlorophyll absorbs wavelengths best at violet, blue, and red, which explains why plants look green, because they don't absorb well in the green area of the visible spectrum. Chlorophyll B is referred to as an accessory pigment or a secondary pigment because it's not directly involved in converting light energy to chemical energy, but it can help absorb wavelengths that chlorophyll A does not, and then use that energy and assist in driving photosynthesis. So looking at our chlorophylls, the general structure of the molecule is here on the right, and we can see right away that there is one major difference right there where this R group is on our entire chlorophyll molecule. It differs by either having this CH3 or methyl group or this CHO group right here, and that's one different. That's the difference in why they absorb differently. They're chlorophyll A and chlorophyll B. But overall, the entire molecule includes a porphyrin ring, which is this ring. This is several rings that have alternating single and double bonds that you can see. Like there's a double bond right there, and then we have a single bond right next to it, and then another double bond right here. And so that's characteristic of it. Also notice that our chlorophyll molecule has a magnesium atom there in the middle. And so what happens here is when light strikes, it gets absorbed by this porphyrin ring, and that excites the electrons in the ring, and then that excitation or that energy gets absorbed and moved around, causing these double bonds and single bonds to alternate with each other. So this double bond might move to the next location, leaving a single bond behind. And overall, that energy gets passed away or passed out of the ring. The original notes kind of originally said that the electrons get shuttled away, but it's not. It's the photons of light that get shuttled. And they may be bounced around and passed from one chlorophyll molecule to another until they get to a very specific chlorophyll A, where the electron is going to be transferred onto a pathway or a chain where those electrons will be used for driving the process of photosynthesis. And so we're looking at the absorption spectrum is actually related to the action spectrum. So the action spectrum would be the spectrum at which we're most efficient at driving a process, for example, photosynthesis. And it actually corresponds to the absorption spectrum as far as the pigments of photosynthesis occur. So you can see right here it does correspond to the absorption spectrum. And a really simple experiment can be done where, if you look on the right here, you see there is an organism that we've seen in the lab before. This is a green algae called Spirogyra, and it does photosynthesis just like green plants does. And then you have some aerobic bacteria there that require oxygen. And so what you do is you shine white light through a prism, and that separates the light into the different wavelengths. And so different wavelengths are shining on different areas of your algae, and you can see where the majority of the bacteria that require oxygen are situated within there. The bacteria move to where there's more oxygen, which they require. So you can see the majority of oxygen are over here, where the action spectrum shows the optimum action or photosynthetic action. And again, it corresponds to the absorption spectrum of those chlorophylls that we see. A couple of other accessory pigments that we see. You may be familiar with carotenoids. You see those in carrots, which look orange. And like the other pigments, these pigments are going to have alternating double and single bond rings that are good for absorbing photons of energy, different wavelengths, other than the chlorophylls. So these carotenoids can actually help assist to absorb wavelengths of light that the chlorophylls do not. So they can increase the range at which we absorb sunlight energy. The other useful thing for carotenoids is they're well known for helping to prevent side reactions that actually destroy molecules. So these carotenoids also help pick up what are called free radicals. Free radicals are the result of other reactions that take place within the cells, and they can be very destructive overall to molecules within the cell. So they're going to be referred to as, if they pick up free radicals, they're referred to as antioxidants, because oxygen actually is quite responsible as being a radical if it's not carefully processed within the cell. So the carotenoids can have this protective role within the cell as well as helping to absorb other wavelengths of light. In aquatic photosynthetic organisms, they may also have these phycobilioproteins, which actually help absorb better in the greens, and the reason for that is if they live in deeper water, a lot of the algae that lives towards the top has already absorbed most of the green light, and I mean most of the violet, blue, and red light, so all that's left is green light. So these phycobilioproteins can help as accessory pigments absorb the light that is available and then pass that light on to the chlorophyll molecules, pass that energy on there to drive photosynthesis. And you can see here, if we answer the question, well, why in fall if you live in places where we have the trees that change color, why do they go from green to orange? Well, that's because the chlorophyll molecules are least stable. So when the leaves begin to die off during the fall, they stop making or maintaining the chlorophyll, and the chlorophyll is the first to degrade. And the carotenoids are more stable, so they stay behind, leaving the leaves to look orange or green or yellow and all these beautiful colors during the fall.